Assalamu alaikum. You are watching Microbiology by Nouman. In this video, we will talk about Streptococci and classification of Streptococci on the basis of hemolysis. Before we start this video, please subscribe my channel and like this video. So let's start. Streptococci are gram-positive cocci. That means after performing gram staining technique, Streptococci bacteria have round shape symmetry and appearing purple in color. So round shape bacteria having purple color after gram staining. A very important test that differentiates Streptococci from Staphylococci is a catalase test. All Streptococci are catalase negative. Whereas all Staphylococci are catalase positive, Streptococci are catalase negative. Also, Streptococci are non-motile and non-spore forming. Streptococci are arranged in the form of chains or in the form of pairs. That means when we see them after gram staining technique, they appear in the form of pairs like this one or in the form of chains. Streptococci are classified on the basis of two categories. The first category on the basis of hemolysis on blood agar plate, and another classification of Streptococci they are classified on the basis of Lancefield classification. But in this video, we will be talking about Streptococci classification on the basis of hemolysis on blood agar plate. Hemolysis means breakdown of red blood cells, and on blood agar plate is a culture media that is commonly used in microbiology, especially for Streptococci. Blood agar plate is used, and some of the Streptococci can completely break down red blood cells from the blood agar plate, whereas certain Streptococci partially break down these red blood cells, and another group, this gamma-hemolytic Streptococci, do not lyse or break red blood cells. We will talk about in detail. So, on the basis of this hemolysis pattern, Streptococci are classified into three groups: alpha-hemolytic Streptococci, beta-hemolytic Streptococci, and gamma-hemolytic Streptococci. First, we'll talk about alpha-hemolytic Streptococci. Alpha-hemolytic Streptococci means when these Streptococci grow on the surface of blood agar plate, they cannot completely break down red blood cells from the blood agar plate. So they causes partial hemolysis or partial breakdown of red blood cells. So alpha hemolysis is partial hemolysis. And a bacteria that produces partial hemolysis, around that bacterial colony, there will be a green zone surrounding the colony. So there will be a greenish zone that indicate the alpha hemolysis. These alpha hemolytic streptococci are further classified into two groups: Streptococcus pneumoniae, which is also known as pneumococcus, and the second group is Viridans Streptococci. This is a group of bacteria that is present in our oral cavity. These two bacteria are separated or differentiated from one another on the basis of certain characteristics. First is an optogen test. Optogen is an antibiotic. Uh, Streptococcus pneumoniae is sensitive to this optogen antibiotic or optogen disc, whereas this Viridans streptococci is resistant to this optogen. So Streptococcus pneumoniae are sensitive. That means if this optogen is present, Streptococcus pneumoniae can't grow. Whereas Viridans streptococci are resistant. That means Viridans streptococci can grow in this presence of this optogen. Second test that differentiate between these two group of bacteria is bile solubility test. This test differentiate between bacteria that are bile soluble and bile insoluble. Streptococcus pneumoniae, if we mix Streptococcus pneumoniae colony with the bile, we see that this bile will be able to dissolve or solubilize the Streptococcus pneumoniae colony and bile will turn transparent. So Streptococcus pneumoniae are bile soluble. Whereas if you mix the colonies of Viridans streptococci with the bile, this bile will not be able to dissolve Viridans streptococci colony. So Viridans streptococci are bile resistant. They are bile soluble. These are insoluble. Third test that differentiate between these two bacteria is a capsule coiling test. This test detects whether a bacteria contain capsule or not. Streptococcus pneumoniae contain polysaccharide capsule, whereas this group do not contain any capsule. So Viridans streptococci are non-capsulated. Streptococcus pneumoniae are capsulated. I already told you Streptococcus pneumoniae are also known as pneumococcus. After gram staining under microscope, they appear in the form of lancet shape. And around this lancet shape, they contain a capsule. So Streptococcus pneumoniae are in short gram-positive cocci, catalase negative, alpha-hemolytic. They have a lancet-shaped appearance. They are sensitive to optogen. They are bile-soluble and contain a capsule. Whereas Viridans streptococci are gram-positive cocci, catalase negative. They produces alpha-hemolysis, so they are alpha-hemolytic bacteria. They are resistant to optogen. They are bile insoluble and non-capsulated. Now talking about the second group of streptococci, which is beta-hemolytic streptococci. Beta-hemolytic or beta-hemolysis means these bacteria can completely break down red blood cells from blood agar plate. So they causes complete hemolysis or complete lysis or complete breakdown of red blood cells. So it's a complete lysis, complete hemolysis. And bacteria that produces beta hemolysis around the bacterial colony, there will be a transparent or yellowish zone that will clearly tell us that it's a beta hemolytic bacteria. Transparent zone. These beta-hemolytic streptococci are further classified into these two groups, Streptococcus pyogenes and Streptococcus agalactiae. This Streptococcus pyogenes is also known as Group A strep, whereas Streptococcus agalactiae is also known as Group B strep. These two groups, Streptococcus pyogenes containing many bacteria that share certain similar characteristics, and similarly this group B strep contain bacteria that share similar characteristics. These two bacteria are separated or differentiated from one another on the basis of basic tracing test. Basic tracing is an antibiotic. This Streptococcus pyogenes is sensitive to basic tracing. That means in the presence of this basic tracing, Streptococcus pyogenes cannot grow. Whereas Streptococcus agalactiae are basic tracing resistant. That means they have no effect of basic tracing on their growth. So if we talk about the characteristics of Streptococcus pyogenes, these are gram-positive cocci, Catalase negative, β-hemolytic. They are also called group A strep, and they are basic tracing sensitive. Whereas Streptococcus agalactiae are gram-positive cocci, Catalase negative, produces beta-hemolysis, so beta-hemolytic Streptococci. They are also called group B strep, and they are basic tracing resistant. Now coming to the third group of Streptococci, which is known as gamma-hemolytic Streptococci. Gamma hemolysis means no hemolysis. That means these bacteria, these Streptococci cannot break down red blood cells completely or partially. They cannot break down red blood cells anyways. So gamma hemolysis is no hemolysis. And these gamma hemolytic Streptococci are further classified on the basis of this property, whether they grow in the presence of 6.5% NaCl, any agar or any broth that contains 6.5% NaCl detect whether these bacteria, gamma hemolytic Streptococci, grow and not. They are further classified into two broad categories. Remember, it's an old classification of this gamma hemolytic Streptococci. It's an old classification. If bacteria do not grow in the presence of 6.5% NaCl, this group, these bacteria belong to Streptococcus bovis. If bacteria, these gamma hemolytic Streptococcus bacteria, grow in the presence of 6.5% NaCl, they are further classified into two groups, Enterococcus faecium and Enterococcus faecalis. In the old classification of Streptococci, Enterococcus were also included under Streptococci. But now Enterococcus have their own genera. They are separated from Streptococci. But this classification is an old classification on the basis of hemolysis, so we are discussing Enterococcus over here. But remember, Enterococci are separated and differentiated from this Streptococci classification. So Enterococcus faecium and Enterococcus faecalis. To differentiate between them, vancomycin is an antibiotic that differentiates between them. Enterococcus faecium are vancomycin resistant. That means in the presence of this vancomycin, Enterococcus faecium bacteria can grow. Whereas Enterococcus faecalis are vancomycin sensitive. That means if vancomycin is present, these bacteria cannot grow. So in short, gamma hemolytic is no hemolysis whether bacteria grow in the presence of 6.5% NaCl. These are two groups are formed. No growth on 6.5% NaCl. These bacteria are Streptococcus bovis that produces gamma hemolysis. They are gram-positive cocci and Catalase negative. Another group is Enterococcus faecium and Enterococcus faecalis. These two are differentiated on the basis of vancomycin, and Enterococci are now classified separately from these Streptococci. That's it from this video. If you have any question regarding this topic, you can ask in the comment section. For more microbiology content, please like this channel and subscribe and like this channel. Thank you.